The polymerase chain reaction, or PCR, is a method by which scientists can amplify DNA, rapidly increasing the copies of a specific DNA sequence in a solution to over 1 billion times the original number. PCR has a wide range of applications, such as obtaining DNA from individuals to diagnose genetic diseases or solve crimes, making multiple copies of a gene to insert the gene into another organism, or providing enough DNA to determine the sequence of a gene. The target DNA is the piece of DNA being amplified in the PCR reaction. After each round of PCR, the amount of target DNA doubles. The deoxyribonucleoside triphosphates, or DNTPs, are the building blocks of the new DNA being polymerized. There are 4 different DNTPs: deoxyadenosine triphosphate, or DATP, deoxyguanosine triphosphate, or DGTP, deoxythymidine triphosphate, or DTTP, and deoxycytidine triphosphate, or DCTP. These DNTPs are covalently linked to the growing DNA strand and provide the energy necessary for the reaction to take place. DNA polymerase is responsible for reading along the strands of the target DNA and covalently linking the complementary nucleotides on the new DNA strands. The polymerase chain reaction uses a DNA polymerase taken from a thermophilic bacterium such as Thermus aquaticus, which can survive the high temperatures of the PCR process. A DNA primer is a short piece of DNA with a nucleotide sequence complementary to one end of the target DNA. These primers are necessary because DNA polymerase can only add bases to the 3' end of a DNA molecule. It cannot start a new DNA molecule from scratch. With each cycle through the polymerase chain reaction, the amount of DNA present doubles. After 30 rounds of PCR, the target DNA has been amplified by more than 1 billion times. During PCR, the temperature is cycled between a high temperature, a relatively low temperature, and an intermediate temperature. The high temperature denatures the DNA, the low temperature allows DNA primers to bind, and the intermediate temperature allows for the rapid replication of DNA. A machine called a thermocycler cycles through these temperatures repeatedly, doubling the amount of DNA present with each cycle. The first step of PCR is denaturation. During this step, the DNA is incubated at a high temperature, which causes the DNA strands to separate by breaking the hydrogen bonds between each base pair. The second step of PCR is priming. During this step, the DNA is incubated at a relatively low temperature, allowing the DNA primers to attach to the single-stranded target DNA. The third step of PCR is extension. During this step, the DNA is incubated at an intermediate temperature, at which the DNA polymerase rapidly replicates the DNA.